We were discussing chapter number 8, and in the previous session we are discussing the structure of prokaryotic cell. We have discussed the few components they are present in prokaryotic cell, like the nucleus that contains a genomic DNA and not covered by a nuclear membrane. It also contains some extracellular DNA that is called as a plasmid, a small circular DNA that you can find outside the genomic DNA. It also contains three envelopes, three layers that forms an envelope. The outermost is glycocalyx, middle is cell wall, the innermost is cell membrane. We have also discussed that from this mesosome there is a formation of cell wall, DNA replication, respiration, and many processes like secretion also takes place. So these are the components, few of the components that takes place. Even the cyanobacteria that also contain a specialized structure called as chromatophore, which contains a pigment, and due to which the bacteria can also do a photosynthesis, but it is only found into a certain bacteria. Okay, so that we have discussed. Now today we are going to discuss the remaining parts of this bacterial cell and the prokaryotic cell. We are discussing this prokaryotic cell with more emphasis on the bacterial cell. Okay, now we discuss the remaining parts. That means what the cytoplasm contains and which are the other extensions that are present in the structure of bacteria. Okay, so let's start with the other components that you can find into a bacterial cell. Another component you can find is a long filamentous structure. Like this is a long filamentous structure like this. Okay, they generates from the envelope. A long filamentous structure look like a tail. Okay, this long filamentous structure that you can find or that arises from the surface of it, that is called as flagella. What it can be called as flagella. You can find this flagella, a long filamentous structure. What is the function of it? This flagella, it is responsible for motility. The flagella is responsible for a motility. That means if the bacteria possess this flagella, then the bacteria are motile. If they don't possess this flagella, the bacteria are non-motile. Motile means movable, can move, and the non-motile means they cannot move. Okay, so based on the presence or absence of flagella, the bacteria can be either motile or it may be non-motile. This flagella consists of three parts. These are the three components you can find into a flagella. I write over here. Which are they? They are filament. Second one is hook. And the third one is basal body. Third one is basal body. So these are the three components you can find into a flagella which are responsible for motility. They are responsible for motility. Clear? So this is about a flagella extension which comes out of the cell wall or the cell envelope. Okay. Now apart from this flagella, the bacteria also possess a small tubular processes that arises from the surface of it. Okay, small tubular tube-like processes which arises from the surface of it. This is called as pili. What it can be called as pili. A small tubular processes arises from the surface of bacteria, that is called as a pili. And the pili has important role in conjugation. In which process? Conjugation. A kind of a process where the genetic material gets exchanged between two bacteria, or it is a kind of a formation of a bridge between two bacteria. That process is called as a conjugation. And conjugation is due to a pili. Okay, a small tubular processes that arises from the surface of a cell. Okay, apart from that, some bacteria also possess a small blister-like structure. These are the small blister-like structure that arises from it. This small blister-like structure are called as fibrin. What it is called as a fibrin. And the function of this fibrin, the fibrin play an important role in attachment. Okay, it's play an important role in attachment. Attachment to the rocks, attachment to a certain surfaces. That attachment of a bacteria to a certain rocks and surfaces, that is due to a presence of small bristle-like structures or small brush-like structures that arises from it, called as a fibrin. So these are the three components that arises from the surface of a cell. The first one is flagella. Okay, this is the first one. This is a flagella. The second one is pili, and the third one is fibrin. Okay, these are the three surfaces which arises from the surface of a bacteria. It is not necessary that all these three surfaces can be found in bacteria. Okay, it may happen that some bacteria possess two, some possess only one, and some also possess three. Okay, so depends on the type of bacteria. There are three processes or extensions that arises from the surface of bacteria: flagella, pili, and fibrin. The major difference between them is flagella, it is a long, slender, filamentous structure and responsible for motility, whereas pili and fibrin, they are the short processes. Pili involved in conjugation process, and the fibrin, it's responsible for attachment to rocks and the surfaces. Okay, so these are the components that arises from the surface of a bacteria. Now we also have to discuss the components that are present into a cytoplasm. This is cytoplasm. That means anything which is present between a nucleus and cell membrane, that we are calling it as cytoplasm. So what is found into a cytoplasm, or what the cytoplasm contain of a bacterial cell? So the cytoplasm of bacterial cell, the one more important thing, what it contains ribosome. Ribosome is the most important content you can find into a cytoplasm of a bacterial cell. And this ribosome, it's made up of two subunits, one large subunit and the another one is small subunit. Okay, there are two subunits. Okay, they form a ribosome. The size of this larger subunit is 50 years. The size of smaller subunit is 30 years. These are the sizes. I again repeat, the size of larger subunit is 50 years. The size of smaller subunit is 30 years. Both the subunits join together and form a total size. The total size is, I am writing over here, the total size is 50 plus 30. It's actually 80 years, but here the size is 70 years. Why 70 years? 50 plus 30 doesn't make us 80, it makes us 70 years. Reason, because when these two subunits that attaches some portion, they gets reduced. So the size gets reduced, hence the size is 70 years. Okay, when they attach, the some portion gets reduced, like this much portion or this much portion that gets reduced. So the size gets reduced, and the size of prokaryotic ribosome or the bacterial ribosome is 70 years, which consists of two subunits, larger and smaller subunit. The size of larger subunit is 50 years, size of smaller subunit is 30 years. Okay, so that is the ribosome you can find into a cytoplasm. But in cytoplasm, where it is found? In a bacterial cell, this ribosome is also attached to a cell membrane. This is a cell membrane. To this cell membrane, these ribosomes are attached. And what is its function? The function of this ribosome, they are involved in protein synthesis. What is the function? They are involved in protein synthesis. Hence, you know that the ribosomes are also considered as factories of protein synthesis, because the process of protein synthesis that occurs on ribosome provides a site and location on which the synthesis of protein takes place. Hence, they are considered as sites of protein synthesis. Okay, so ribosomes are also considered as factories of protein synthesis. In prokaryotic cell or the ribosome, many times one mRNA that attaches with a many ribosome, or you can say a reverse one. Many ribosomes that attaches to a single mRNA. There is only a one mRNA. But to this mRNA, many ribosomes are attached in a chain, one by one. First, second, third, fourth. Okay, the mRNA is a single one, and to this mRNA, many ribosomes gets attached. And this kind of a condition, formation of this kind of a condition is called as polysome formation. Polysome formation. Or you can also call it as polyribosome formation. It's called as polysome formation or polyribosome formation. So what is polysome formation? When many ribosome attaches to a single mRNA and form a chain-like structure, that's called as polysome or polyribosome formation. Okay, so that is about the ribosome. I again repeat, what is a ribosome? Ribosomes, they are present on the cell membrane, made up of two subunits, larger, smaller. The size of larger subunit is 50 S, size of smaller subunit is 30 S, total size is 70 S. S means it's a unit, it's a Swedenborg unit. Okay. Ribosomes are considered as factories of protein synthesis because it provides a site and location for a protein synthesis, where protein synthesis takes place. In prokaryotic cell, mostly many ribosomes attaches to a single mRNA and form polysome or polyribosome formation. That's about the component you can find in a cytoplasm of a bacterial cell. The another component you can find into a cytoplasm is inclusion bodies. Which one is a inclusion bodies? Okay. So what this inclusion body contains? Another component you can find is inclusion body. Inclusion bodies, plural one, inclusion bodies. Okay. What this inclusion body contain? The inclusion body contain reserve material. It contains a reserve material. Reserve material may be in the form of food, or it may be in the form of nutrient, or it may be in the form of you can say waste. Okay, so in any form, the reserve food material that is found into a specialized structure called as inclusion bodies. Okay, this inclusion body, they are going to be named based on the material it stores. Like for an example, I will give you an example that will more clarify. Like for an example, phosphate granule. Phosphate granule. Okay, so this is a name of inclusion body, phosphate granule. What it store? It store a phosphate. Okay. Second one is, it's a easy one. The glycogen granule. The glycogen granule. What it store? It store glycogen. Okay. Another one is cyanophycean granule. So now you know that what it contains. It contains cyanophycean. So ultimate thing is that they are going to name based on the material what they store. Like for an example, phosphate granule, what it store? Phosphate. Glycogen granule, what it store? Glycogen. Cyanophycean granule, they are stored cyanophycean, a kind of a pigment. Okay, so based on the material they store or the reserve material they possess, they are going to name. Okay, one more thing you can find, that is a gas vacuole. Write it down and then write. The another inclusion body you can find, but that is not found in every bacteria. The another one is gas vacuole. This is another kind of a inclusion body. The name itself suggests obviously what it stores. It contains a gas. But this kind of a gas vacuole is not found in every bacteria. They are found in specific type of bacteria. So which bacteria possess this gas vacuole? These gas vacuoles are reported in blue-green, green and purple photosynthetic bacteria. It's found in blue-green, green and purple photosynthetic bacteria. What they possess? They possess gas vacuole. Okay, gas vacuole is not found in every bacteria. Okay, so that's about a gas vacuole. So I can repeat, what is an inclusion body? Inclusion bodies are the structure that is found into a cytoplasm, like here. Okay, here. They are non-membranous because the membranous structures are not found in prokaryotic cell or bacterial cell. They are the non-membranous structure. Okay, they store a reserve food material, and based on the reserve food material they are going to be named. Like for an example, phosphate granule, glycogen granule, etc. Similarly, some bacteria or few bacteria possess gas vacuole which store gas, and they can be found in blue-green, green and purple photosynthetic bacteria. Okay, so this is about inclusion bodies, and here we finish with prokaryotic cell. Okay, now we have to start with eukaryotic cell. We have already seen the difference between prokaryotic cell and the eukaryotic cell. Okay, so what is the difference? The prokaryotic cells are smaller, eukaryotic are larger. Prokaryotic divides fast, eukaryotic divides slow. In prokaryotic cell, well-organized nucleus is absent. In case of eukaryotic cell, there is a presence of well-organized nucleus. In case of a prokaryotic cell, membranous structures are absent. In case of a eukaryotic cell, the membranous structures are present. The examples what we have discussed, and we have also seen the components. But now we have to see some introduction of eukaryotic cell, and then in the next session we will learn the components you can find in a eukaryotic cell. Okay, so let's start the eukaryotic cell. Here we finish with the prokaryotic cell and we start with eukaryotic cell. Just we will do an introduction of eukaryotic cell today. So by comparing the characteristics of eukaryotic cell with prokaryotic cell, you already know it, the characteristic of this cell, eukaryotic cell. So I just enlist again what are the characteristics we have already discussed during the comparison of or difference between the prokaryotic and eukaryotic cell, but here just I list out which are the characteristics you can find in only eukaryotic cells. The first characteristic you can find, they have well organized nucleus. So well organized nucleus is a characteristic of eukaryotic cell. The second one is they possess or they have membrane bound organelles. They have membrane bound organelles like ER, Golgi body, lysosome, vacuole, mitochondria, microbodies that you can find in a eukaryotic cell. They are absent in prokaryotic cell. They are relatively, means comparatively, larger, and comparatively they divide slowly. Okay, and these are the characteristics of the basic characteristic of eukaryotic cell. The examples are fungi, plants, animals. These are the usual examples you can find in eukaryotic cell: fungi, plants and animals. But now the question comes, all these eukaryotic cells are same like the plant cell and animal cell? We are animal and the plant. Thus the plant cell and the animal cell, they are same. So what are the differences between the plants and the animal cell? That we have to discuss. So now the one point is clear, that all the type of eukaryotic cells are not same, but they share some basic characteristic. These are the basic characteristic you can find in every eukaryotic cell, but all the eukaryotic cells are not same. We have to most focus on these two type of a cell, plant cell and the animal cell. So let's see what are the differences that exist between the plant and the animal cell. So just see the difference between plant cell and the second one is animal cell. The first difference in a plant cell, there is a presence of cell wall. So I write down cell wall is present. Okay, in animal cell you cannot find a cell wall. So cell wall is absent. Okay, you have already learned in the cell theory that the animal cell has a single layer around them, and that is cell membrane. They don't possess a cell wall. Cell wall is absent. But yes, plant cell possesses a cell wall, and even the bacterial cell also possesses a cell wall. So cell wall is found in a plant cell, but it is absent in animal cell. This is the first difference. Second one is plastid. The plastid is present in plant cell, but that plastid is absent in animal cell. Plastid is found in plant cell, it is absent in animal cell. Plastid means, I give you one example of plastid, chloroplast. Chloroplast is an example of a plastid. So that chloroplast can be found in a plant cell, but can you find this chloroplast into animal cell? No. The chloroplast cannot be found in an animal cell. Okay, so that plastids are present in plant cell, they are absent in animal cell. The third difference, the plant cell, they have large vacuole. Okay, they have large vacuole, but the animal cell, they don't have large vacuole. Large vacuole is absent in animal cell. Okay, so this is third characteristic or the third difference between the plant and the animal cell. So in short, what it wants to say that cell wall, plastid and large vacuole are found in plant cell, but cell wall, plastid and large vacuole are absent in animal cell. You cannot find these three components in animal cell which can be found in plant cell. Now the another difference between the plant and the animal cell, you know that in case of a plant cell, centriole is absent. This is a non-membrane bound organelle, and you know that non-membrane bound organelles are found in both prokaryotic and eukaryotic cell. Okay, but that centriole is absent in plant cell, and you can find this centriole in animal cell. Okay, so the centriole can be found in animal cell. Remember, centriole is only found in animal cell. They are absent in plant cell. It has an important role during the cell division. In short, these are the differences. Cell wall, plastid and large vacuole are found in plant cell or present in plant cell. They are absent in animal cell. The reverse, centriole is absent in plant cell, but it is present into an animal cell. So if you have been asked the organelle that can only be found in animal cell, the name is centriole. And if you have been asked the organelles that is only found in a plant cell but cannot be found in animal cell, the organelles are cell wall, plastids and large vacuole. So that's all about the differences between the two types of eukaryotic cell, plant and the animal cell. So students, today just we have discussed The remaining part of prokaryotic cell like flagella, pili, fibrin, ribosome, inclusion bodies, and we have seen the common characteristic of eukaryotic cell with its example, and we have also differentiated between the two types of eukaryotic cell, that are plant and the animal cell. In next session, we will learn in detail the structural components you find into an animal cell. So we discussed each and every component you can find into a eukaryotic cell in the next session. Till then, keep watching, keep learning. Don't forget to like and subscribe this channel. Like and subscribe this channel. Thank you for watching.